My name is Paula Grammas, and I'm the inaugural director of the George and Ann Ryan Institute for Neuroscience at the University of Rhode Island. I was recruited here to establish the Ryan Institute for Neuroscience, and the idea was that we were interested in looking at the causes of neurodegenerative diseases as a category, and really, obviously, Alzheimer's is kind of the primary one. We have really made an effort as we recruit new faculty here and build the Institute to bring people here who think outside the box, who want to come in and collaborate with us to further explore our interests in the vasculature, but also bring complementary interests, people that have expertise in neuroimmunology, inflammation, and are approaching Alzheimer's disease from lots of different angles, because I think ultimately that's how we're going to be successful. My lab has worked for many years and looking at the role that cerebral blood vessels play in the development of Alzheimer's disease. We have really been among the pioneers to look at a vascular, potentially a blood vessel connection to the death of nerve cells or neurons in the Alzheimer brain. Alzheimer's disease, as you know, is characterized by nerve cell death. So the $64,000 question is how that comes to be. One of the pathways that we think is important and really underexplored is the role that blood vessels play in this process. We developed a technology years ago for isolating blood vessels from the brain and applied it to looking at human postmortem material to understand biochemically how the Alzheimer brain is different from patients that have died that are the same age but that don't have Alzheimer's disease. And we showed in a series of studies over a number of years that these vessels become what we call pathologically activated and release toxic factors that directly kill nerve cells. The many risk factors for potentially developing Alzheimer's disease are associated with cardiovascular alterations. We know that midlife high cholesterol is correlated with an increased risk for late life dementia, Alzheimer's disease. Midlife hypertension correlates with the development of late life dementia. And we know that these cardiovascular risk factors have a direct impact on blood vessels. The question is how these risk factors translate their properties in the blood vessel so that it becomes altered and releases the factors. That's actually the project that we're funded now by the Cure Alzheimer's Fund to look at. Ultimately, we want to look for a disease-modifying therapy. All the drugs that are on the market are what we call symptom-modifying drugs, meaning that they prolong some quality of life for a period of time, but they ultimately don't get at what's happening in the Alzheimer brain, which is nerve cell death. So from our perspective, having identified the fact that toxic factors come out of these vessels and kill neurons, we think opens up lots of different therapeutic options. We have, in some experimental animal models, we've been able to show that a few different types of drugs can block that activation pathway. And by blocking it, we've shown that we can diminish release of toxic factors, which in the animal models is very similar to what we see in human material. And interestingly, in the animal models, when you block the release of these toxic factors, you also improve cognitive function, because the animal models also show the same things that humans show. They show memory deterioration with age. We think that's really exciting because that says, again, that this process likely is involved in some aspect of the development of Alzheimer's disease. It's been sort of an uphill battle because there's been a lot of focus and a lot of research has been in amyloid. But I think now, with so many decades of sort of failed approaches, people are more willing to think a little bit more broadly and also accept this idea that Alzheimer's disease is likely not a single disorder, but a group of diseases that in the end have a common clinical manifestation, dementia, and a common pathology, but multiple etiologies. And at least some of that, in my opinion, is driven by blood vessel dysfunction. We were very appreciative of receiving funding from the Cure Alzheimer's Fund because the work that we do here at the Ryan Institute and in my laboratory is looking at a target that's really hasn't been explored. The vascular connection in Alzheimer's disease, particularly from the way that we're looking at it, this idea that there's a synthetic metabolic defect in brain blood vessels that helps drive Alzheimer's pathology, has really not been looked at. And as you may know, getting funding for ideas that are outside the box from national agencies like NIH are actually really hard. So being able to have funding to look at underexplored areas, new targets, out-of-the-box thinking, I think is really exciting, and we're very appreciative of being able to do that.